what I'm going to try and do is pull together some of the key elements, the take-homes, looking also at the critical issue of dosage. And during the bits and pieces that we're going to be covering, I also want us to think about some of the language that we use in talking about nucleotides, because we really are looking at the emergence of a new element, a very important and extremely fundamental element in nutritional science. And what I've noticed over the last few years is that it's difficult enough for medical doctors especially, but even nutritional therapists, to understand why is it that we weren't taught this when we were doing our nutrition courses, for example. And so, you know, we know that you are an advanced audience. I can see that just by going out there and looking at the fact that when you have a pot of walnuts and raisins and seeds, the walnuts is easily the most consumed, and that's not just because they look like little brains, but it's because you know what's in them. You barely touch the raisins, and you don't overdo it on the seeds because you're worried about pushing up your omega-6, omega-3 ratio. So, and in many respects, you imagine even that level of understanding is very different from the people out of this room. And so we need to find models of communication that help people to understand that nutrition is still at a very, very early stage of development. We have people in universities who believe that they know an awful lot about it. The very fact that it was only 100 years or so ago that we were looking at things like vitamins and minerals. In fact, the precursors to today's drug industry were busy researching and developing, if you like, synthetic analogues of vitamins and minerals that are the bulk of vitamins and minerals we now see in our supermarkets and pharmacies that are not natural at all. We're only just now discovering 100 years later that when we deliver them, like drugs in isolated forms, they don't work in the way that they do in nature, which is why we see problems with high-dose vitamin E, problems with high-dose synthetic folate, problems with high-dose beta-carotene. So we're very, very clumsy in terms of how we understand these issues. And, you know, we've heard about the fact that we are bioelectrical beings. We have to look at the fact that over the next few hundred years, we're going to look at this whole issue in a very different way. One of the things we really understand about nucleotides is they are absolutely fundamental. So these are some of the issues we're going to look at. Even if you look at the macronutrient categories, we understand very little about some of these categories. We don't, for example, fully understand. We used to think 20 years ago that proteins were like Meccano sets in which you could just assemble amino acids and deliver them. We didn't mind if they were folded by high temperature cooking. We didn't think that taking a piece of sirloin steak and frying it into a crisp with polyaromatic hydrocarbons was any different from what the Japanese were doing, eating their raw sashimi. And then, in fact, we thought we were one step better because we weren't poisoning ourselves with mercury. But then if you look at what we're now beginning to learn about some of these macronutrient categories, we realize we're still infants in the classroom. There's still an incredible amount to learn. The public have just about come to terms with the fact that there are different types of carbohydrate and different types of fat. We use long terms like healthy fats and unhealthy fats, and we use complex carbohydrates and simple carbohydrates to explain the speed at which they're broken up into glucose when we consume them. We have a debate going on about obesity that I've got to say I spend my life scratching my head thinking about how primitive that debate is when the people who lead the debate are still talking about input and output, energy expenditure and energy use, when the whole thing is about an extraordinarily complex dance between our environment and our genes. And most of what we can learn about it is actually in our gene bank and history, ancestral history. And so we are learning now about things like low levels of cyclic ketosis as an important mechanism that stop when you carry on eating, as the dietitians tell us to do, every 3 hours or 2 hours in order to stabilize blood sugar levels. When you get onto some of the micronutrients, it kind of stops after you talk about vitamins, minerals, and amino acids. And so there are numerous people who've not learnt anything formally about nucleotides. Biochemists certainly have, and of course, one of the ways in which we can look at this is imagine a really sophisticated computer in which you had a keyboard, a 4-letter alphabet keyboard, that you had the 4 nucleotide bases in your keyboard that you could tap out. That information then gets communicated through your processor. And of course, nucleotides are part of the keyboard and they're part of that processor, because as they basically engage in the process of self-replication within the nucleus of the cell, and the swapping out between the thymine and the uracil happens in the RNA strand, and the RNA then is transcribed and shifts out of the nucleus and into the ribosome, where you get the transcription, the linking, the formation of peptides, and then the production of amino acids and proteins. You basically have the printer of your computer engaged. It's printing out those amino acids and those proteins from the ribosomes. So it's a kind of computer system that doesn't involve this sort of solid-state compartment that uses limited electrical impulses. The thing actually moves through the system, from the nucleus to the ribosomes and then into the extracellular space. It's a very, very complex system. And of course, it started 4.5 billion years ago, ostensibly. And of course, there's many ways of looking at how it happened, whether we're looking at the seeding hypothesis, where these organic molecules, self-replicating organic molecules, came in from outer space. And there's plenty of evidence they do exist there, or whether bolts of lightning and incredible reactions occurred with the presence of cyanide that actually kicked the process off. But organic particles don't get more fundamental than nucleotides. We've learned that they basically have these 3 components. We've got a pentose sugar, either ribose in RNA or deoxyribose in DNA. We've got the bases, which can include inosine, which is also a purine. And where do we find lots of inosine? We find it in certain foods, and it's very associated with the umami flavor, you know, that delicious flavor. We talked about chicken broths and broths being a rich source of nucleotides. If you look at fermented soya products, you produce a lot of inosine compounds in there. And that delicious umami flavor that you get with tamari, for example, or a chicken broth, that is because you have a very high inosine component. And of course, in terms of the degradation pathway, that goes straight down the purine catabolism pathway and ends up with xanthine, which degrades to uric acid. And uric acid, we think of it as a terrible thing because for people who are susceptible or producing a lot of it, it forms uric acid crystals, which of course is gout. But let's remember that the body isn't that silly. Something as fundamental as this is producing uric acid, which at the levels that are lower than harmful levels actually is a really important antioxidant. So therein lies some of the complexities of things that can be harmful at one dose and very beneficial at other doses. So you really have got this idea that there are three major pathways: de novo synthesis, salvage or dietary intake. You have heard probably more than 20 times today about conditional essentiality. I think in my last slide, I'm going to pose the question whether we are actually talking about a conditionally essential nutrient, or now what is becoming an essential nutrient. And in fact, what we've just heard, the very fact that people like to stay on the products, the very fact that people benefit and have a higher level of health, is an indication that actually they need that additional resource because of the amount of gut dysbiosis we have around, the amount of stress that we have around, the dysfunction in the gut microbiome that we have around, and the fact, very, very importantly, that we now consume so little exogenous nucleotides in our general diets. In terms of the nucleotide pool, we are beginning to have some understanding about certain elements of it. Most of the research has been on the adenine pool, and you can understand why that's so critical because of ATP production for energy. We have much less information on some of the other pools. It's not one single pool. It's still got the attachment to the 4 key nitrogenous bases, and it's got different numbers of phosphates attached to it, which of course can be shifted around very easily. But very fundamentally, if you become deficient in one particular base, you have a problem. That's a very fundamental requirement. We're only at the early stages of beginning to understand that. So here we have the output from the degradation pathway for the purines. And what's interesting is the pyrimidines, the very group that are deficient in our diets, as you've seen earlier, and we'll see some more information on that, produce a whole bunch of beta amino acids that are very beneficial for us. And now we're beginning to see, again in the sports area, and I look over there because I see 2 really sporting people, that beta-alanine, carnosine, and we've heard about Now Foods, they sell those products to athletes like they're going out of fashion, are incredibly beneficial. These are outputs of the pyrimidine pathway. So if you don't have enough pyrimidine in the system, you don't produce some of these products that are beneficial for people who are stressing their body to the limits. And time for a very brief anecdote. You know, I do spend quite a lot of time looking at trials, but I'm a great believer, particularly when you're talking about an area of emerging science. One of the most powerful things we can do in helping people understand the importance of this category of nutrients is to tell stories, which is what we've just heard. My own story is very simple. I'm a keen cyclist, and what I've had is a very long-term problem. When I push my body and ride in excess of 160 kilometers, 100 miles or so, I will get a cold sore. It's an indication that my immune system is not coping with the latent herpes simplex virus and the cold sore expresses, and I have to hide for a few days. But I've now done it. I think I'm, as of the 200-miler to Paris, it's now 19 times. I'm almost hitting 20 times. This anecdote has been repeated, where I have not had expression of a cold sore on the condition that I get up to the kind of doses I'm going to talk about in a minute, before I do the ride and after I do the ride. It is, you know, if we think about, as therapists, the combination of vitamin C and zinc, for example, we've been worshipping at that altar for many years. These are in a different category. Vitamin C and zinc on their own will have no impact on my cold sore expression. So it's N equals 1, but there is now 19 replications of that event, and it goes along with the kind of fundamental nature of how important these things are for immune function. So this was a critical paper that came out, I remember, the excitement that Peter had when he saw it coming out in Cell back in 2011. And it is a recognition that if the nucleotide pool is suppressed, a whole bunch of the most fundamental processes that we require for healthy life, including genome stability, the very process that becomes unstable when we see the development of tumors in the body, the DNA repair, again, that's absolutely vital, and the production of cancer, that does not happen properly where you have a deficient nucleotide pool. So we're at the beginning of really beginning to understand that process. In terms of what they're doing, well, they are kind of magic molecules. They're doing a lot more than just feeding in that 4-letter alphabet program that initiates transcription and translation and production of new cells. So that is the nucleic acid biosynthesis that is so critical. But they are really important in energy production because they act as critical components in the production of ATP, but also the transduction, the movement of that through various enzymes that are required to shift it from one part of the body to another. Critically, obviously, in terms of the production of protein in ribosomes, because of the production of the importance of very small peptide and kinase molecules in communication processes. They're absolutely vital to that. You cannot function adequately without sufficient nucleotides, and a whole range of other biomolecules. Anything that is required to be transcribed by those particular segments of DNA, active segments, structural segments that produce proteins. So completely and utterly vital for life. Now, we're also beginning to see, we've talked about pyrimidine nucleotides being limited. E. coli is a bacterial model for which there's been a lot of research. We're now beginning to see papers that are showing that if you don't have sufficient thymine within the system, you actually get death. And that is a system that is being explored now in E. coli. It's always been there. And in fact, there may be mechanisms that are inbuilt in the senescence process that we now think of as being very much related to telomeres, where actually certain nitrogenous bases become less available and actually cause an organism to die out. We can also see very basic fundamental work going on with organisms as interesting as sea cucumbers. And in this case, we've got a sea cucumber that is challenged by a very common marine bacteria, Vibrio splendidus. And in this case, what we're seeing is if you deliver a very high dose of nucleotides, and let's get this into some perspective. This is a level that's about 180 times than the maximum level the European authorities, and you did hear I was going to talk something about regulation, but is 180 times higher than the maximum level that the European authorities allow you to put into that ridiculous cow's milk formula that they insist on giving babies. And that level is, again, a minimum threshold. So, in fact, in this particular study, they looked at 2 levels, and lo and behold, the highest level was the level that delivered the best result in terms of the immune response that this sea cucumber has to this bacteria. So, looking at your point on dosage, there really isn't too high a dose if you are not delivering just the purine component. All of the safety assessments that are looked at are based on excessive intakes of purines that actually create hyperuricemia, high urate levels within the bloodstream that could, in susceptible individuals produce gout, and then they use a whole range of safety factors to get you down to these ridiculously low levels that stave away disease but don't take you anywhere near the kind of levels you need for optimal health. And also what you begin to see that if you start stressing a system, this is looking at Saccharomyces cerevisiae, baker's yeast, brewer's yeast, and you use, in this case, a very light, empty medium with very little glucose in it, and then another medium with a little bit more glucose, you start to see a subtle variation in terms of the nucleotide levels, the ATP production, but most importantly, how these relate to viability. And you can see, just don't forget the time axis here is to 12 days, there it's to 30 days. And what's interesting here is that the viability is actually related to the stress, and it can be measured by the inadequate amount of nucleotides available because you're in a very limited nutrient broth. Again, if you'd stressed it with low sugar and you'd put it in a nucleotide-rich environment, you don't get this result. So this equates to the people out there who are having very bad diets, very little, very few nucleotides within the diet, and putting themselves under immense stress and not having enough resources for the immune system to function properly. So we recognize that the colostrum, and this is again, it shows you how early we are in the emergence of this science. There is absolute recognition in the 1970s, and we've done some really interesting work with Professor Angel Gil down at Granada University, who's one of the key scientists who was doing all the fundamental research that really forced the infant formula companies to start to add nucleotides to their products. And they'll say, yes, if you don't put the nucleotides in the breast milk, and it's now become compulsory, in the infant formulas, up to the first 6 months, it's illegal because the babies are being starved of nutrients. Never mind the incorrect balance of amino acids and proteins and everything else in there, all the synthetic vitamins. But there are mothers who can't breastfeed. There are far too many who are not encouraged on how to get through mastitis and other issues, and that's another story. In terms of supplemental nucleotides, there has been an understanding that for these so-called conditionally essential groups, there will be people who are under children who are in rapid growth and also have immune challenge or GI infections and so on. So again, these are extreme cases. And so for many years, until Peter came along, it was thought that if you're a healthy adult, you don't need it. You don't need any additional support. Let's just focus briefly. We've talked about, when we do spend a lot of time thinking about the human body and the human cell components, but let's not forget the microbiome itself that contains 10 times more cells, 100 trillion cells, 10 times more cells than we have human cells in the entire body, has a desperate need, if it's going to function properly, to have sufficient nucleotides. And you are delivering them, particularly in supplemental form, directly to the point where they can be incorporated into the nucleus of cells and produce new bacterial cells that we need for our health, to be able to have a robust immune reaction, to be able to process foods properly, to reduce our risk of autoimmune diseases. A lot of autoimmune diseases are caused by complete instability in the gut microbiome, and we think we need to take probiotics, but actually, again, when we take nucleotides, we're doing something that's much more fundamental. We're feeding the system that has this extraordinary, it's not just the gut mucosa that has such a rapid rate of cell reproduction, it is the microbiome itself. So the turnover is extremely rapid. And obviously the lymphoid tissue itself, highly mobile, incredible numbers of cells in order to allow it to function. Again, the requirement ramps up massively during disease, stress, any rapid growth. We're appreciating at least conditional essentiality. We've got to understand when you're going to be in a period of peak requirement. I think as clinicians that's absolutely key, and that's why I think Rachel asked the question, did you reduce the dose after that? So this concept of having a high dose at times of peak requirement and then a maintenance dose makes absolute sense, particularly in terms of the cost of buying the supplemental product. It makes sense to everyone, and it makes scientific sense. So obviously the immune system, the GI system, absolutely critical for that. Now we get to the EU and what they've done. Now, first thing to notice is these are the exclusive ribonucleotides that they have said are suitable for use in infant formulae. So Nestlé and a whole bunch of people came to a decision. They found a regulatory home in a particular EU directive for this limited group. And it's quite interesting. If you think of nucleotides generally, they do have different functions. Have you heard of monosodium glutamate, for example? You know, it is essentially a nucleotide. And of course, again, in different forms, it has different effects. There are studies that have been done in Australia when I was living there that showed actually the highest levels of MSG were in Italian restaurants, not Chinese restaurants. And that's because of the amount of naturally occurring MSG that comes from sun-dried tomatoes, concentration of tomatoes. And again, gives that very nice flavor that you taste so readily. Something happens when Ajinomoto and companies start tampering with the synthetic version of it, and it behaves in a different way. But bottom line is we've got, you'll see the inosine form there, you've got the two purine forms, and then we've got two pyrimidine forms up at the top here. So, and of course inosine is a purine. So it's a limited range, and frankly, from what we have been learning about ancestral intakes, if we assume that this was enough for a baby based on measurements from breast milk, they are very low levels once you translate them to an adult that is much larger and may also be in immune system distress, for example. But we'll look at that in a minute. So again, you'll see the kind of levels we're looking at. This is in milligrams per day. So an infant of 1 month of age as opposed to 12 months who's trying to grow requires the highest intake. So this is 6 milligrams a day. Just compare that against the protein requirement, for example. What do you all... There's views that many people will look at somewhere between 0.7 of a gram and 1.2 grams of protein. So it's a small amount compared with the amount of protein. And obviously we've also had Leach do a major study looking at total potentially available nucleosides. So that's the nucleoside is the nucleotide minus the phosphate group. And again, you'll see slightly higher levels, but that was one of the ways in which they came to their conclusions. You'll also see, again, different studies have looked at different groups of nucleotide, and you'll see we have limited information. We generally see a consistent lack of information on pyrimidine nucleotides, a little bit more on the crucial purine ones, but they don't tend to be in a deficient state. The critical thing here is to see when you look at infant formula and what the infant formula companies have been doing, you'll see basically the kind of levels per 100 grams that look at about 23 milligrams per 100 grams, or per 100 calories, about 4.5. So again, fairly small amounts. And magically, this infant formula is meant to turn into something that equates to breast milk, when breast milk actually is changing all the time between individuals at different times of day, according to what's being eaten, etc., etc. Peter has obviously done an awful lot of work in developing the understanding of the importance of supplemental nucleotides by looking at animal feeds. So when you look at aquaculture or poultry or pig farming, where you have a high intensity of farming and really stressed animals, often living off very limited diets, what is incredibly relevant here is that when you start adding supplemental nucleotides, suddenly you can reduce or even eliminate the need for antibiotics. What does that tell us? Again, how important they are. So, again, depending on what feed is being given to the animal, there are huge variations in the amount of nucleotides that are being received. And one of the big changes we've seen in animal feed over the last 10 years or so is this massive shift to genetically modified soya and maize, which are, and soya is very low in nucleotides unless it's fermented. And of course, when it's fermented, it's largely because the bacteria are making such a significant contribution to it. When we look at meats, that's where we start to get the really high levels. So, you know, if you do indulge in, say, liver pate, you can get a lot of it, or raw liver, if that's your particular cup of tea. You'll see tofu here fermented, so it's, you know, this is 200 milligrams per typical standard portion, you know, a fillet steak is not doing too badly here, but you'll see how you can get quite easily to a kind of minimal level of about 500 milligrams from the diet. So, and of course, there are individuals on particular days who might be getting much lower than that, who may be getting only 100 milligrams, depending on what they're eating. So it can be very, very variable. And we start, if we look at various fruits and vegetables, the levels, as we've heard, are very low. If you look at dairy products or dairy substitutes, even soya milk, you can see, is still pretty low. So bottom line is, if you're not eating this kind of stuff, which most people aren't, your levels will be very, very low. And what we're suggesting is that when you have a conditional requirement for peak intake, that the level will probably be in the low gram level, okay, 1 to 2 grams, 1,000 to 2,000 milligrams. This slide is probably the most important summation of a lot of work that I did with Professor Angel Gil, that these are estimates. We had to, for the European authorities, come up with an ADI, an average daily intake. So we had to do assessments looking at a lot of data. And EFSA have basically accepted this because Angel Gil was very much involved in the early work, and there aren't a lot of data outside the data that Peter's been collecting and that are referenced in the other dietary assessments. But in essence, if you're consuming 500 milligrams per day, you have eaten one of the typical higher sources in that day. The problem is, if you look at a whole week or a month or a year, you are maybe eating those sources once, twice, 3 times a week at most. So it's a bit like the oily fish argument when you're looking at omega-3, omega-6 balance. If you look at our ancestral diets, well, definitely we were consuming in the low gram range. with about 3,000 milligrams perhaps being about an average, that equates to 50 milligrams per kilogram. And one of the difficulties in terms of the way the regulators see this is they say, well, all of these nucleotides are the same, so we're going to look at the ones that produce higher levels of uric acid, in other words, the purine forms, and we're going to then use conversion factors. And in essence, what we can all agree is that even looking only at purine forms, you can definitely consume 33 milligrams per kilogram body weight. 33 kilograms. If anyone has a calculator, I think that equates to about 1,800 milligrams per day for a 60-kilogram adult. So that immediately takes you into the 1 to 2 gram range, and that lowest observable adverse effect level is based on hyperuricemia, elevated uric acid levels that could lead in sensitive people. So for most of us, and if you're using a pyrimidine-dominant product, you can safely double that. Yeah? You can safely double that level and then get yourself back to ancestral levels. For people on vegetarian diets, it's even more important to be taking significant amounts. So in order to be on the super safe side, and you might decide to go outside this kind of range when you're within clinic, but these sort of figures that we're all comfortable about talking about more publicly, and it's also thinking about the ramifications of these products being considered medicines by regulators. We know that we're in the high end of the physiological range if we start talking about 25 to 50 milligrams per kilogram body weight. Okay? So those are the kind of levels that you should be comfortable with. And I'm just going to finish off briefly to show you where we are from a regulatory perspective. Here's the food medicine division in Europe. What happens is they compartmentalize everything in terms of ring fences. So you've got to be in a particular ring fence to be legal. If you fall outside it, you become illegal. Clearly, taking products from food as extract means that they can exist as foods. As soon as you concentrate them and put them into a supplemental form, they become food supplements. However, the ingredients themselves are still existing, but only for a short time till this Parnut system eventually falls over and is abolished on the 20th of July next year in this group that we call Foods for Particular Nutritional Uses, or Parnuts. And that's how the infant formula varieties have been used. What we have to be wary of is not to fall into either the novel food camp, the two red areas, or the drug camp. You can be a novel food by saying that you are even if you are naturally derived, if you deliver your nutrient in a unique range of compositions that is not found in the normal food supply, you could be classified as a novel food. So again, you need to be careful about not talking about extreme levels of pyrimidine that are completely different from anything that you can find in the diet, because you'll find someone like me, in order to save the risk of it being classified as medicine, will be finding all the possible ways in which you could actually manage in a normal diet to get high pyrimidine levels in, which is not easy. It's relatively safe. What you have to be very careful of, obviously, is not claiming medicinal effects, but that applies for any nutrient. So on that note, I'll just summarize by saying maybe we shouldn't be thinking of these nutrients anymore as conditionally essential nutrients. We should probably be aiming for upwards of 750 milligrams per day supplemental intake using a balanced blend. It's code for using the nucleotide form. Obviously, the real benefits are in periods of rapid growth, for gut integrity, for the microbiome, for the immune system, for exercise stress, for recovery. You've got to look at, as Jonathan so pointed out so well, all the cofactors that are needed to make sure that the whole system can function properly. So if you're looking at folate, I'd probably disagree with your 800 microgram levels unless you get it from synthetic forms, but I'd strongly suggest you use methylated folate, and then you can easily go up even to EFSA's safest level of 1,800 micrograms per day of methylated folate, either the glucosamine or the calcium methylfolate forms, methylated B12, methylcobalamin, B6, magnesium, zinc, and for women, usually not for men, additional iron. And obviously do all the other things that you're also good at doing in terms of reducing other sources of stress and getting the diet right. So on that note, thank you very much indeed.